Okay, hopefully everyone is in. There's still a couple of people joining, but that's fine. We'll get started. So welcome everyone to today's webinar, and thanks very much for your time and interest on this topic. So my name's Claudia, and joining me remotely today from Sydney is my colleague Lisa. So this will be a 30-minute session covering Randox's range of quality control solutions for infectious serology testing. As some housekeeping, we do have the microphones on mute, but if you have a question throughout the session, please enter it in the chat box and we'll try to get to as many of these as possible at the end. Now, this session's also being recorded, so a copy will be made available to everyone at the end, as well as those who had registered for the session but have not been able to attend, and you're then welcome to forward this on to your friends and colleagues. So yes, we'll get started. So many of you are familiar with Randox, but here is a brief overview of who we are and where we've come from. So Randox was established in 1982, just outside of Belfast in Northern Ireland. Today, we manufacture over 4.1 billion diagnostic tests annually, and we estimate that that goes on to help diagnose over 370 million people. Randox played a major role in the COVID-19 pandemic. We quickly had to sort of restructure our workforces to perform over 26 million COVID-19 tests. But today we supply over 100,000 laboratories globally, and lots of you, I'm sure, help make up this number. In terms of our workforce, we have over 2,500 employees across over 145 countries, and 800 of our employees are dedicated R&D scientists and engineers who help make the products and services what they are. So Randox Holding Group consists of several different product categories, all the way from health clinics of our own in the UK, the US, France, and to more routine laboratory products that we manufacture, like reagents, clinical chemistry analyzers, multiplex immunoassay instruments, and what we're here to talk about today, our quality control solutions. So today we'll go through our three key areas of quality control portfolio in the context of infectious serology. So firstly, we'll go through our Accusera range of internal quality control materials. So these are our third-party negative and positive controls for serology to help ensure the accuracy and consistency of your results in every run. Then we'll go through Accusera 24/7, so how you can interface or manually input your QC data onto our cloud-based QC data management software to track and monitor your QC results, but then also compare your QC results with the Randox peer group network around the world. So Lisa will show you on the screen some screenshots of Accusera 24/7 and how easy it is to go in, review your Levy-Jennings charts, have staff members leave comments for you to follow up on, gather your statistics and generate reports. And then we'll finish off with RICAS. So RICAS is Randox's external quality assessment scheme. It's the largest quality assessment scheme in the world now, and we'll go through what we have on offer for infectious serology testing and the peer numbers that come along with this. So we'll pass on to Lisa now to take you through the internal quality control sections.
Thank you, Claudia. So as Claudia just introduced, I'm going to give an overview of serology-focused IQC products in our Accusera range. So our serology quality controls are designed to deliver a cost-effective and high-quality solution for the routine performance monitoring of a variety of infectious diseases. So firstly, I'll give you a bit of an overview of the key benefits of the Randox QCs. So these cover a wide range of infectious diseases and are compatible with use on some of the most popular immunoassay platforms. Our serology QCs all have an extended open vial stability, which ensures that waste is kept to a minimum. They're all manufactured using 100% human serum, so this helps to minimise any matrix effects. And they're conveniently supplied in a liquid ready-to-use format, so no preparation is required in the lab. We have a range of multi-analyte controls available, which reduces the number of individual controls required to cover your full test menu. And additionally, all of our serology controls have gained CE and TGA marking. So now to run through the controls that we do offer. Firstly, we have our Epstein-Barr virus QC. So this one QC includes nuclear antigen IgG and viral capsid antigen IgG and viral capsid antigen IgM. Next, moving on to our serology controls. So these are split into 3 levels of serology positive, as well as an extremely consolidated serology negative. So we have our serology 1 positive, serology 2 positive, and serology 3 positive. And these controls comprise infectious diseases including HIV, hepatitis, HTLV, and syphilis. Then, as you can see, the negatives are all consolidated into one control, so there's no need to purchase 3 separately. So just one negative QC to cover all of your tests. Then moving on to our torch controls. Now our torch control range comprises positive controls for both IgG and IgM antibodies, and again, one negative control to cover all. So firstly, we have our torch IgG positive control, our torch IgM positive control, and then again, one negative control to cover all tests. So this control consolidates multiple analytes, including CMV, EBV, HSV1 and 2. And then this also includes measles, mumps, and rubella, minimizing the need for multiple controls. Finally, we have our specific proteins control as part of our immunology QCs. So this control covers 26 analytes and is available in 3 clinically relevant levels. So most notably for our serology area, this includes strep or ASO, as well as rheumatoid factor and kappa and lambda light chains. So just to summarise again, the key benefits of our controls are they cover a wide range of infectious diseases. They all have a long open vial stability period. They're manufactured using 100% human serum. They're liquid ready to use for convenience in the lab, and they're all multi-analyte, so we can consolidate your testing down to just a small number of vials. So now moving on to our Accusera 24/7 software, as Claudia also introduced. So we all should know that in line with ISO 15189, data should be recorded so that trends and shifts are detectable, and statistical techniques should be applied to this data. However, monitoring of QC data can be extremely complex and time-consuming within the lab. So this is where Accusera 24/7 comes in. So Accusera 24/7 is our live cloud-based data management and peer group reporting software. It's intended to assist you in the lab with your management of daily QC activities. The cloud-based feature of this lab means you can access the software anywhere that you have internet, and this removes the need for any complex installation. So firstly, to highlight the key features of Accusera 24/7. It has a really unique dashboard interface, which is easy to use and reduces time spent analysing data. It's updated in live real time, so labs can compare statistics to their peer groups and identify any issues to reduce time on troubleshooting. It automatically calculates advanced statistics such as percentage bias, total error, and sigma metrics. It has the ability to monitor and compare performance and assess trends with automatically generated charts, including Levy-Jennings charts. It generates comprehensive and customizable reports to speed up your review process. You can manage and compare the performance of multiple labs from multiple departments in one site to multiple national sites within one lab chain. It's really flexible, so it allows you to configure your own performance limits, multi-rules, and target values for each instrument or lot of QC. And finally, you can upload QC data directly to 24/7 via LIMS or middleware, eliminating the need for manual entry. However, this option is available for anyone who needs that. So now I'll take you through a brief demo of the software in relation to your serology QCs. So first of all, we have our homepage, and you can view all of your data on the dashboard for fast and easy access to your results. So we like to call this our navigational hub. We have simple icon-based interface with user-friendly design, and as you can see, all of your analytes are in one place. And a newer feature of this is that you can easily search by analyte, chemistry type, or method. So for example, in this screenshot, we've searched by serology, and you can view all of your serology tests in one place. So from this hub, you can select any analyte you'd like to look at and click these three dots on the right-hand side. And this will show you your results history, charts, or QC test configuration. So first of all, we have the results history page. So here you can view all QC results entered for a particular test. So the results are really conveniently color-coded. We have red for a reject and orange for an alert. And you can also filter this page at the top, so in this little box here, to view alerted or rejected QCs, or to view any QCs that have comments attached. So this helps ensure quick and easy performance assessment. On this results history page, you can also then generate your statistics. So as you can see, we have monthly and cumulative statistics, which are automatically calculated and displayed for each lot of control. So these include basic statistics such as mean, standard deviation, percentage CV, as well as your percentage bias, total error, and sigma score. Now moving on to our charts page. So here you have the ability to monitor performance and trends with automatically generated Levy-Jennings charts. So on your LG charts, you can view the summary of events to better visualize events which may be affecting QC performance. So for example, on this screen, you can see we have a Randox QC, preventative maintenance, or a reagent lot change. So you can see how any of these events might be affecting your QC performance. Now each comment can be added to any point and reviewed by managers to help with traceability. And these individual values can be differentiated by different icons, as we've demonstrated with the key at the bottom here. So you can easily tell if a value has been accepted or rejected and if there's a comment attached. You can also combine multiple instruments, analytes, or QC lots onto one single chart, and this can be toggled on and off at the bottom for easier visualization. We then also have the ability to combine and overlay multiple QCs onto one chart to help better understand the performance of maybe first-party versus third-party QCs. So for example, here you can see a lab who went from using a first-party serology QC on the left to a third-party serology QC on the right. And again, lots can be toggled on and off with this little feature at the bottom to visualize each separately. From your charts page, you can also use the icons along the top here to choose to view your performance summary. So this provides a visual assessment of accuracy and precision in relation to your chosen peer group. So for example, you can easily assess the performance of your lab with your peer group, in this case for Rubella IgG QC. So I've explained how you can view this data on our dashboard for fast and easy access to your results, but you can use 24/7 to delve a little bit deeper and to access comprehensive, easy-to-read reports. So there's quite a few reports that are really useful, and I won't go through all of them, but a few notable ones are the statistical reports, the uncertainty of measurement report, the exception report, and the peer group report. So first of all, for the statistical report, this allows you to view your IQC data from a specified date range and compare it to your cumulative data as well as the peer group data, all within one screen. Importantly, as I mentioned before, if you're part of a chain of labs, you can select the group icon at the top, where you can compare this data with your lab group. This means that you can easily identify any labs in your group falling away from the group mean. And like all of our reports and charts, this data is fully exportable to PDF or Excel with these icons at the top for filing or for data review. Another really useful report is the uncertainty of measurement report. So ISO guidelines require for each of its quantitative tests, a lab must have measurement of uncertainty values that it can provide if requested. So our software provides automatic calculation of measurement of uncertainty, saving valuable time and help you meeting your ISO requirements with ease. If you would like to determine your best and worst performing QCs, our exception report is perfect for this. So this report is designed to quickly identify QCs with a high percentage of errors. So as you can see, this provides an on-screen summary of the number of QC results for each lot that fall within 2 standard deviations, 2 to 3 standard deviations, or over 3 standard deviations. So what's really handy about this is that this performance review can be filtered to display the worst performing QCs at the top by just clicking on this 3 SDI column. So this is extremely useful when it comes to reviewing QC performance in preparation for an audit. The final report I want to show you is the peer group statistics report. So this is updated in live and real time with no submission deadlines. So here you can compare your statistics to those of your peer group determined by analyte, method, instrument manufacturer, or model. So what you can do here is simply select the QC lot you wish to analyse at the top in these boxes here, and AccuSare 24/7 will just generate the data for you. So this gives you really great insight into your performance versus your peers. So that's a brief overview of how AccuSare 24/7 can assist in management of your serology QC data. Now I'm just going to pass over to Claudia to discuss our RICUS programs.
Okay, thanks, Lisa, for taking us through the internal quality control. So this last section will be on external proficiency testing for serology tests. So the Randox QAP scheme is known as RICUS. RICUS is an acronym, so it stands for Randox International Quality Assessment Scheme. There are many external quality assessment schemes out there, but there are a couple of things that makes RICUS so special. So the first of which is our global reach. RICUS is now the world's largest EQA scheme. We have over 75,000 participants across 139 countries. So this means a lot of data and a lot of peers for you to compare your results against. We do have a flexible portfolio, so not just our serology programs, but we offer 37 highly consolidated programs, which means that we have a variety of options to cover a wide test menu. Importantly, RICUS is a UCAS accredited proficiency testing provider, and we are accredited to the ISO standard 17043, which is the conformity assessment for proficiency testing providers. So RICUS, it operates very similar to many other quality assurance schemes. However, there are a couple of key differences. So firstly, you can register at any time, either for the full program or for pro rata. And pro rata can be a really excellent way to trial the program for a couple of months before deciding if it's right for your laboratory and enrolling in a full year's program. The other key difference is with registration, you can enroll up to 5 instruments. So this can be instruments of the same make and model that run the same tests, or this can be instruments of different makes and models that run a different subset of tests. And this is at no extra cost, even for the reporting ability of your results. So after your enrollment, Randox will dispatch your samples, and these are numbered. Some programs, like our serology programs, must be manufactured and dispatched in halves. And this is because they are 100% real human-based material and liquid ready to use. You'll receive your login details to the online RICUSnet portal, where you will enter your results before the monthly or fortnightly deadline. And then you will receive your RICUS reports. So another benefit of RICUS is at the turnaround time of reports. The serology report turnaround time is about 10 days after the final submission date. So you'll get this report as a PDF to your email, and it will also be available on the RICUSnet portal. And this quicker turnaround time allows you to take any corrective action faster if necessary. At the end of your cycle, you will receive a certificate of participation and an end of cycle summary report. So I understand many of you already participate in some RICUS programs, but we'll go through a serology report format. So the serology report formats are set out slightly differently to the normal RICUS. As you can see, they are still 1 page per parameter. We just offer you 1 page of consolidated data for you to review per parameter. But as you can see here, the left side here is for your qualitative result comparison, and the right side of your report is for quantitative comparison. And for each serology parameter, you can choose whether you want to report qualitatively, quantitatively, or both. And there is no extra cost associated with reporting either or both. So if we zoom into the top left section of the report, you'll see here, this is your test name and the units that you're enrolled in. And here you'll see a breakdown of your peers. So the top row shows you all method statistics. This is everybody enrolled in that parameter in this program. And then below, it will show you your method group statistics, so only those enrolled in your method category. So this will show you a summary of results, with N being your number of peers, your mean, the CV percent of the results from that peer group, uncertainty of the mean, SDPA, which stands for Standard Deviation of Performance Assessment. So this is similar to SD, however, it's calculated taking various other factors into consideration, factors that are necessary for our accreditation as a proficiency testing provider. And it will also show you the number of participants that had their results excluded in this sample as outliers. So then in the box below, the black triangle will indicate your result. So it will give you what you reported as quantitative and what you reported as qualitative if you are reporting both, and show the mean for comparison. Then to the right, you'll see these two metrics here, SDI and RMSDI. So SDI is Standard Deviation Index, and this is an indication of how far your reported result is from the consensus mean, from your method mean peer group, relative to the SDPA. And RICAS, in all programs, not just the serology programs, they class an acceptable performance as an SDI less than plus or minus 2. And then below SDI, you'll see RMSDI, which just stands for Running Mean Standard Deviation Index. And this is a metric that calculates the average of your last 10 SDIs. And this can be quite a useful metric to track your performance over time. So like I mentioned, on the left-hand side of the serology report, this is your qualitative reporting section. So this will show the consensus acceptable result for your method group for that sample. And there is a nice clear histogram here to show how many participants have reported this sample as non-reactive, how many reported it as inconclusive, and how many reported it as reactive. Now this green shading highlights here your method group. And in the serology programs, it is really important to only compare with your method group, because there are often different cutoff points for different assays, and these are sometimes not interchangeable. But we do have a multi-method stat section below this. So this allows you to have a look at the results from other method groups if you'd like to. Then on the right-hand side of the page is the quantitative section. So here you will have your Levy-Jennings chart with the last 20 samples plot as your SDIs. So this can be a really great way to track your performance and visualize any sudden shifts or trends over time. And we would always recommend, especially if your analyzer does not collect this information, to note down the reagent batch and calibrator batch details at the time of testing each RICA sample, and even things like how long the reagent or calibrator has been open for, because issues that we quite often see in these sort of graphs, of issues that you can visualize in this sort of Levy-Jennings chart, can be due to a calibrator or reagent losing its stability or something of that nature. So then again, here below, you can see the histogram. So this shows you all method peers in white, and then the green highlights your own method peers. And the black triangle here indicates where you are on this histogram. And then again below, you can see your multi-method stat section. So this is other methods who are enrolled in the program. If you participate in RICAS and are needing some support to interpret your RICAS results, there are some great resources available. So we do have one resource called the Evaluation of Performance Guide, and that is found in the RICASnet portal at the bottom of the homepage once you log in. Otherwise, we are certainly available to speak with individually, or the other option is host group sessions for your laboratory. So along with the report side of RICAS, another one of our aims is to help maximise efficiency in the lab. And we do this by consolidating our RICAS programs and tests in the programs. So there's no need for several individual programs now for all of your serology tests, that may look something like this, when you can consolidate them into just 4 RICAS programs. And so we'll talk about each. So first one here is our HIV hepatitis program. This has 2 samples to report on every month. So it's not fortnightly because both samples are due on the same day each month. They're 1.8 mil samples, liquid ready to use, and 100% human-based material. The official start dates here of this program are January or July, and you can see here the parameters included, as well as some peer groups. Now, this peer group list is non-exhaustive. These are just some peer groups that could be relevant to your laboratory. But as Randox RICAS program is the largest global QAP scheme in the world, we often have peer group categories that some other proficiency testing schemes don't have. So we have a good range of older instruments, newer instruments, smaller throughput, higher throughput, etc. If you can't find a peer group in another program, it's likely that you will find it in RICAS. So secondly, our torch program. So this again is 2 samples every month. The samples are 1 mil liquid ready to use, and the start date again is January or July. And you can see here that similar to our QC, the RICAS program consolidates the IgGs and IgMs together, even mumps, measles, and rubella. So you don't need a separate program. These are all included in the one sample. And you can see an overview of our peer groups here. Our EBV program, so this is again 1 sample, instead this is 1 sample per month. It's 1 mil liquid ready to use, start date in January or July, and we cover the 3 EBV parameters. And syphilis, this is 1 sample per month, 1 mil liquid ready to use, and starting in January or July. Now this program is suitable for various methods. If you if you do screening tests or you do confirmatory testing for syphilis, whether that is by immunoassay, RPR, VDRL, or TPHA methods, this syphilis the program is suitable for all of these methods. We've just listed here the immunoassay peer groups, but you can also request the titer method peer groups from us as well. And lastly, we wanted to mention our specific proteins program. So this program, you can choose between a fortnightly or a monthly submission. And this has 26 different protein parameters, including ASO. And remember, with RICAS, you can enroll up to 5 instruments free of charge. So if you wanted to share this program across multiple analyzers or across different departments, you can absolutely do so. And our peer group for ASO is growing. We now have 211 participants. Okay, so this brings us to the end of our session. If you liked anything that you saw here today, here are the relevant contacts to reach out to. If you're not sure, the customer support email and phone is open to anybody, so you may send any queries in here. We have a couple of minutes for questions and answers. If anybody has any questions, I've just had a look here in the chat box. There's been nothing written, but if anybody has any questions, you're welcome to write them here, or else email one of us and we can get back to you individually. But otherwise, thanks very much for joining, everyone. And we hope you have a good rest of your day, and the recording will be made available to you by email shortly.